Women in India, many of these housewives, they develop a large amount of, biceps and triceps and they become very fat. Okay, despite they working, at homes. That's a typical, type 2 diabetes mellitus phenotype. Actually, is there a common polymorphism seen across those individuals? The answer is no. So you find you find at least 60 to 70 mutations that are different, and not every mutation is seen in those particular individuals that are common to this particular phenotype. Despite having almost 1 million human genomes that are publicly available today, still not under the confluence of integrated systems genomic approaches and say that okay, this is the only mutation that is largely seen for, say, for cancers or for diabetes. I have a fair intuition that at a point of time you might expect a gene that might be overexpressed, and that gene that is overexpressed might harbor a kind of a mutation sitting in them, and that particular mutation could be deleterious in nature. Okay, and you'll be very much surprised to know that BRCA1 or BRCA2 mutations that you have, none of the mutations are a result of the gene that is overexpressed in that individual. But I'm sure over the list of some 20,000 and odd genes, there might be some genes that might be overexpressed and yet harbor those particular mutations that are responsible for pathogenesis. So, through this podcast, what I wanted to tell the audience here is that there is a hope for winning a Nobel Prize in the near future for such kind of discoveries that are made. That yes, there are mutations that are happening just because, not for the heck of a reason that these particular mutations are seen or segregated in those particular individuals, but also just because these particular mutations harbored in that particular gene is probably overexpressed or maybe underexpressed as well. Almost 1 crore of America's genomes have been sequenced. 3% of the American genome is sequenced, and yet we are not able to ascertain which individual might be at a risk at this point of time.
This year, 2024, we see Nobel Prize, getting to Victor and Gary for microRNA and its probable role in gene regulation.
As I told you, I started my PhD career as a protein biologist working on hypothetical proteins, and then we discovered a large amount of microRNAs way back then, because some of those microRNAs were annotated as hypothetical proteins, and eventually we annotated those candidates as pseudogenes. They not only identified those LET4, LET14 mechanisms individually, and also they could drive the mechanistic evidence saying that okay, these are those particular candidates that are probably regulatory in nature.
Welcome to another very interesting episode of the podcast. Welcome to the Somnath Show. In this podcast, we have our guest as Dr. Prashanth. Dr. Prashanth N. Suravajhala is Associate Professor in School of Biotechnology, Amrita, and he has an extensive background in systems genomics. In this podcast, we will be understanding what is systems biology. In systems biology, can we address the problems or understand better the rare diseases? What systems biology has an important contribution in deciphering the complicated scenario of rare diseases, and how it is different from the traditional genotypic studies or traditional genetic studies which we have been following for some time. In systems biology also we'll be understanding what is a top-down approach, how holistically we can see the entire mechanism which is going on inside an organism, and in precisely we get a better in-depth analysis of our problems. So these problems can be diseases, these problems can be other genetic mutations, and how these mutations are actually regulating the expression of different genes. So stay tuned, don't go anywhere, because we are going to have a very in-depth understanding of systems genomics. With this, let's welcome Dr. Prashanth. When we talk of bioinformatics as a tool, how one person can use it in different disease models, because all the disease models are very different. So how bioinformatics can play a very important tool in understanding the the pathways maybe, or the phenomena, or the phenotypic expressions of different diseases, what you are talking about tool.
Right, so yeah, that's indeed a great question. Yeah, I mean, I would always say that bioinformatics is a tool. So at any given point of time, you expect that bioinformatics as a tool can lessen the scale of experimentation. Okay, so and of course, albeit the fact that you have to perform those particular what you call annotations or those particular things across 2 or 3 tools, reach consensus, and then come to a conclusion that yes, okay, these are those particular bioinformatics tools that are yielding similar results, and probably this is how the results can be taken further. So as far as the animal models are concerned, I mean, that is probably a secondary approach. So first, we will simply go for an orthology model, which means you expect a particular protein might be with some interactions, and you expect those particular orthologs sitting across those individuals. Then you ask, okay, if this is the protein that has been with some known function, you transfer this knowledge to the other one. So as far as the animal models are concerned, I am bit I would say wary of using animal models, because animal models doesn't honor the 3 R's, because the 3 R's of experimentation should be negated, because I don't see a big animal model discoveries that have been translated to real frame in the humans, with due respect to them. I would probably take it the other way around. And thanks to the AlphaFold technologies that has been done so far today, thanks to the Baker's Rosetta method that has been introduced, for discovery of structural morphs or structural prediction. So we can probably honor those kind of tools, and of course take the AI-based models to a greater hope, and reach consensus. We don't say that okay, we are simply using these bioinformatics tools, but we'll probably develop with a greater simulative scale and come up with a conclusion. And yes, if need be, of course, there are some organizations like CDSCO, a Clinical Drug Safety Compliance Organization. They demand the experiments to be done in order to ensure that the drug comes to the market. So for that particular thing, at least instead of sacrificing those umpteen number of animals at a later point of time, you can probably sacrifice only a few animals, taking the bioinformatics models, at a greater level. So at least we are honoring science to a greater commitment at this point of time.
Very true, very true. Totally agree with that. Okay, as you mentioned about artificial intelligence, these days artificial intelligence has entered our life in a strong way, and in every areas we go, starting from simple mobile phone, maybe it is also integrated with AI. So do you think AI has significantly empowered bioinformatics as a tool?
I'm personally not a big fan of artificial intelligence, because artificial intelligence is a hackneyed term. It has really come up over the years, although it has really come up in different ways. In fact, the good old natural algorithms that have been conceptualized by we humans have been put forth in a better way. Yes, AI can be used, it can be generative, it can be non-generative, but it should not be the sole purpose of our research to take up that particular thing. That is my point. So we can definitely use those particular AI models, but AI models alone is not the realm of taking up bioinformatics research further. That is my point.
Right, right. As I have seen your extensive work on systems biology, I have a very interesting question maybe to start with.
Yeah, please.
Why
Please,
system biology, and why you have chosen this field precisely?
I've been always fascinated to understand the biology of a system, especially the human systems. I always, compared, the system, to any kind of, tools that I would probably work on. So once my 10th standard, my biology teacher started with a wonderful term that, we have about 13 trillion cells, to be precise, about 13.3 trillion cells. And then, with a curiosity in mind, we would ask certain questions, how these 13.3 trillion cells, could really make up. And then, of course, there was nothing like a systems biology that was conceptualized during those heydays. But, I thought that I would definitely take up medicine. Unfortunately, I couldn't take up medicine because I was short of that particular rank. And then my interest went on towards, taking up microbiology as a major during my BSc. And then I took up MSc Biotechnology. And then that was when, the bioinformatics paraphernalia was growing, leaps and bounds. And that was where, I was really fascinated about understanding the biology of system. And of course, I was very poor in wet lab, not just because I was not good at wet lab, but, in our school, we were not that fortunate, to have, the kind of, wet lab oriented practicals. So, so that was how, I was really fascinated with the biology of system, biology of a human system. So as a human biologist, I developed interest in systems biology in that way. So, yeah.
Wow, wow. I think, systems biology, in my early days, I also worked some part of it, but could not take it forward. That's a different story. But in simple terms, what is systems biology or system genomics, to be precise, because you are working on that?
Yeah. So, I mean, systems biology is simply the biology of a system. So, understanding how, the life conceptualizes in a tiny cell to a larger, constituent of, those tissues. Because if you take up any system like a human system, you've got about several tissues, which are of those major components like, whatever body parts that we see. And each of these particular tissues or major components make up those particular cells. And those cells have got, in a eukaryotic system, you've got a nucleus and you've got about 16 other different organelles where there might be these, biomolecules that are localized. So because the proteins represent the expressibility of this particular genome, or it represents the phenotype, so you would probably conceptualize systems biology in terms of how these particular proteins, they keep on interacting with each other. So that is, a typical systems biology. Whereas systems genomics is, omics is nothing but many. So whenever you say om, that is nothing but, many. So genome is nothing but, study of many number of genes. Proteome, study of many number of, proteins. So systems genomics largely encapsulates what amount of, polymorphisms are seen within those particular, cellular organelles or within those particular cells. If we really take about the humans, we have got about 3.2 billion bases of human genome, and every sixth base randomly is really undergoing mutation or polymorphism between you and me or across the humans. But not necessarily every sixth base that is undergoing that kind of polymorphism or change would be associated with deleteriosity. So you would probably expect at any certain point of time that there are mutations that are harmful and there are mutations that are probably beneficial. So that is where, systems genomics is all about. So that is how, I got, interest in systems genomics. I started my career as a protein biologist, and then, when the next generation sequencing came in, I developed interest in systems genomics. Yeah.
Okay. Now, coming to, maybe more precisely, because genomics, when you talk, it is a very wide term. So, right. As far as targeted to a particular cause, maybe the disease factor, or how you are addressing to problem solving of rare diseases, understanding its proliferation, maybe understanding its progression or its cell cycle mechanism from which pathway it is happening. So in what way, system genomics gives you an insight into a disease which is not much understood as of now?
Right. So one of the major entities that we always look is, to understand, what function a kind of a gene entails, So if we really talk about central dogma of molecular biology, we have DNA transcripts, mRNA translates to protein. So when you talk about central dogma of what do you call bioinformatics, we talk about any of these three biomolecules of interest, be it DNA, RNA, and protein, and then you take up those particular sequences, and then we tend to understand if these particular sequences they fold. So primarily, if the protein folds, you got those pockets at acidity, and then you will be able to know what are those particular functions associated with those particular uh, protein structures. So now comes a question. Okay, if we have any kind of mutations at the interface of those particular residues or those particular amino acid residues, where a drug can really bind to those particular regions. So if we really talk at the phenotypic level, there are some diseases that are common, there are diseases that are probably rare, but the common certainty is if there is a mutation that is ascertained for that particular disease or that particular phenotype, and at the interfacial site of that particular mutation, if that particular amino acid is being targeted by any drug or any particular ligand at that particular site. So that is the main question that we would probably ask, taking the central dogma of bioinformatics: sequence predicts structure, structure predicts function. So if that residue is probably well known, and if that residue is targeted by any particular ligand, then we'll probably ask a question, okay, that is the target of interest, and if that particular residue is probably at the hidden site of that particular structure, or if it is exposed. Many a time these particular residues they might be probably hidden, they are not really exposed, and that is the reason why many of these particular phenotypes are less understood and they are far away from these particular therapeutics. Yeah.
Okay.
Yeah.
So in what terms it is then different from the traditional type of genetic testing which we have been doing it?
Yeah, even the genetic testing is also nevertheless the same thing, but you don't really dig at the structural level. Whereas in systems genomics, you dig deeper, you ask a question, okay, if this particular amino acid residue is seen, because if you have seen that amino acid residue, of course, it is going to be a missense or a nonsense mutation, or even a kind of a non-synonymous mutation that you tend to see, which means the nucleotide change is resulting in the change of an amino acid, and then you expect that particular change in amino acid is giving rise to a completely different protein function, and probably that is the reason why there is a disease that is probably caused in that particular person. So when in a normal genetic testing, you know what are the mutations that are commonly seen, what are those particular mutations that are probably seen across those individuals, and then you ask a question, okay, these mutations are seen just because these have been tested across wide population, and over a period of time some of these particular mutations remain as pathogenic over the years, and they might turn out from pathogenic condition to benign condition in terms of some conditions like diabetes, or there might be, in a rare case, there might be certain benign mutations that might eventually turn out to be pathogenic state or pathogenic condition. So in genetic testing, you only know, say for example, okay, I know some hemoglobinopathies like a GS or a GB, and I know a mutation over there, and that is usually a monogenic disorder. Say a sickler would also always have a mutation sitting in that particular chromosome. So that's a monogenic condition. Whereas there are other rare diseases or even some diabetes, from a Jiva's point of view, you don't expect the same mutation that is seen in individuals that are specific to say Indian phenotype. So you might expect a different kind of mutation that is probably polymorphic or disease associated to some other individuals. So the best example that I could attribute to is when I returned back to India in 2016, we worked on a very important phenotype in diabetes called Asian Indian type 2 diabetes mellitus. so which we call it as AIT2DM. So in that, if you see the women in India, they're born, they're born normal, but when they get married, many of these housewives, they develop a large amount of, biceps and triceps, and they become very fat. Okay, despite they working, at homes. That's a typical, type 2 diabetes mellitus phenotype. Actually, it's an obese this thing. But if you see and ask, okay, is there a common polymorphism seen across those individuals? The answer is no. Okay, so you find you find at least 60 to 70 mutations that are different, and not every mutation is seen in those particular individuals that are common to this particular phenotype. Okay, and that begs us a question whether or not the mutations are seen in the non-coding regions as well, because the non-coding regions that are sitting in the intronic chunks, they essentially regulate how the exonic chunks are, are influencing the phenotype. So that's where the systems genomics, in my humble opinion, can really play a very important role. Okay, so this is a classical example that I can correlate the difference between a normal genetic testing and how systems genomics can really be integrated, for rare diseases, for example. Yeah.
Very true, very true. I think that this example is very relevant. We see it, in Indian population. But I have one maybe a very silly question. From cohort to cohort, then these mutations will vary, right? So,
Right.
is this has a relevance with a cohort or a ethnicity or like a population?
Yeah, very
from
much. That's a wonderful question. That's where everyone is really breaking their heads these days. So people do not understand that. Okay, albeit the fact that there have been many guidelines that have been formulated, like American College, of Medical Genetics, Association of Molecular Pathology, American College of Genetic Sciences, and even we have some Indian norms. You have got some guidelines. There are some guidelines like KVIA guidelines. There are some guidelines like Sherlock guidelines that have been introduced, for ascertaining whether or not a mutation that has been described as pathogenic remains as pathogenic. But albeit the fact that there has been two important things, genetic risk scores that have been introduced. One is what we call it as a PRS, which is a polygenic risk score. So which means you expect a mutation if that mutation is seen across large number of populations, and if the large number of populations remain slightly different, you don't expect that mutation to be pathogenic in nature across those individuals, which means it might be just for the heck of a mutation that you discover that it's a mutation, but you don't expect that particular change that you see in the phenotype. That is number one. And number two, okay, then you ask, okay, this is a mutation that is probably commonly discovered, and you expect that this particular mutation might invariably be seen across those individuals in that particular phenotype across the segregated population. So that's the question that you are going to ask. So that is, what you have it for something what we call it as a polygenic risk score. The second thing is what we call it as a Mendelian randomization. Okay, so this has been a kind of a thing, that has largely been known, for identifying whether or not a mutation that is at risk for pathogenesis, which means at any given point of time, a mutation that is conferred as a pathogenic variant over a period of time has eventually turned out to be benign, or a vice versa, which many a time we see. But Mendelian randomization or even polygenic risk score is largely dependent on how that particular individual is attributed to the kind of drug intake he or she does at any given point of time. Okay, so even for a particular disease, if a person is given that particular drug, is that particular drug inducing that kind of state of pathogenesis over a period of time? Okay, so probably that is one of the reasons why a Mendelian randomization or polygenic risk score has been a big failure in diabetes, especially in Indian population. So now that begs us a question, okay, is there a chance that through integrated systems genomic approach, we need to keep on sequencing as many genomes? In fact, that is also not an ideal solution, because sequencing as many genomes is also not a kind of an ideal solution. then even if we sequence, then what amount of genetic variation is sitting in the non-coding regions? That is the kind of question people have asked. People have even identified the genetic variation sitting in the non-coding regions. They have identified the mutation sitting in the say microRNAs or long non-coding RNAs or competitively endogenous RNAs or even pvRNAs. In vain, they could attribute the pathogenesis or risk score to that particular phenotype. Okay, so which means there might be a large number of mutations sitting across the 3.2 billion bases of the human genome, where relatively several of those particular mutations are working in tandem. So which means sometimes you expect some mutation or set of mutations that are acting or regulating all through the point of time at one instance, or there might be mutations. Hey, look, I'm going to cause this particular disease in this particular individual at this particular time scale. Okay, and then the other mutation would say, okay, now that you have caused this particular phenotype in this particular individual, I'll be at a latent phase, so I'll be occurring or I'll be causing that particular change at a later point of time. So they have that kind of maybe they have that kind of division of labor as well, in causing the pathogenesis. Okay,
But
so
what is the factor which influences them mostly?
the most important factor is the epigenetics, how the environmental change influences this particular things, and that is the reason why you don't expect any same phenotype to be the same mutation to be associated with the same phenotype at any given point of time, even if it turns out to be pathogenic to non-pathogenic. So having said this, that is the reason why, despite having almost 1 million human genomes that are publicly available today, okay, we are still not under the confluence of integrated systems genomic approaches and say that, okay, this is a mutation that is largely seen, or this is the only mutation that is largely seen for, say, for cancers or for diabetes or this thing. So again, they are trying to go for something. Today we have something what we call it as a low pass sequencing. So in a low pass sequencing, all the data is not yet publicly available, it is coming up. So people would do the whole genome sequencing, researchers would do the whole genome sequencing with 0.5x, 0.75x, 1x, 1.25x and 1.75x depth of coverage, and see if there are multiple or multitude number of those genomic variants that are segregated across the 3.2 GB data. Yeah, so that is number one. And number two, what our lab and what I as an individual, what I'm very much interested is, we have been working across several of these particular things now over the last 800 exomes that I've personally analyzed. I have a fair intuition that at a point of time you might expect a gene that might be overexpressed, and that gene that is overexpressed might harbor a kind of a mutation sitting in them, and that particular mutation could be deleterious in nature actually. So I'll just give you a classical example, which unfortunately doesn't work. There is a well-known gene called BRCA2. BRCA2 causes not only the mutations seen in breast cancer, but also in cervical cancer, bladder cancer, and of course CA prostate cancer as well. And there are 50 well-known missense mutations that are seen in BRCA. Okay, and you'll be very much surprised to know that the BRCA2 mutations that you have, BRCA1 or BRCA2 mutations that you have, okay, none of the mutations are a result of the gene that is overexpressed in that individual. Yeah, so which means they are abrupt. So there's no cross-link between a mutation that is seen in that individual and a gene that is probably overexpressed. So that begs us a question. I mean, this is from a result of, say, analyzing some n number of genomes. Okay, but I'm sure over the list of some 20,000 and odd genes, there might be some genes that might be overexpressed and yet harbor those particular mutations that are responsible for pathogenesis in those individuals. Okay, so I mean, through this podcast, what I wanted to tell the audience here is that there is a hope for winning a Nobel Prize in the near future for such kind of discoveries that are made, that yes, there are mutations that are happening just because, not for the heck of a reason that these particular mutations are seen or segregated in those particular individuals, but also just because these particular mutations harbored in that particular gene is probably overexpressed or maybe underexpressed as well. Okay, so that is the research that we are going through for the next 10 years. I think there's a hope, for that, and that is what our lab is trying to do, for a number of, organism. I mean, the human genomes, that we are trying to do, at this point of time,
Yeah, for last couple of years, we have also heard, personalized medicine.
True.
And with this, I think personalized medicine or personalized treatments can also be streamlined.
Yes, to a certain extent, Som. I mean, I'll put it the other way around. I mean, personalized medicine, pharmacogenomics has been going around over the last, I would say, 15 years now. Ever since almost the first human genome project, came in and fully came up after 2001 to 2006 or 7. But I would say that the personalized medicine would probably act when the individual can make up a very healthy lifestyle, okay, even after a disease is seen in that particular individual. Okay, so what is really happening here is we are either probably taking a large amount of drugs, and these amount of drugs are probably making that particular disease a kind of a status quo. And that is probably the reason, why we are not able to honor that kind of, precision medicine or personalized medicine. Okay, and even the elite countries, even the developed countries, Americas, boast to have sequenced as many as, 1.5 to 2 million individuals so far, out of 34 crores. Just imagine the population, almost 1 crore of America's genomes have been sequenced. And yet we are not able to ascertain, 3% of the American genome is sequenced, and yet we are not able to ascertain which individual might be at a risk at this point of time. So I mean, if I would probably softly target the doctors, with due respect to them. Doctors, of course, they are the best souls. We consider them to be gods and demigods.
True.
In India, at least, with due respect to them, there are some kind of doctors who are probably commercial, who don't probably consider a patient as from a Hippocratic oath angle, but he or she would probably expect the patient to come again and again, to visit them. So, to ensure that, I mean, with due respect to them, I don't say that all doctors are like this, and at the same time, the onus is also on the patient to ensure that they don't take or they're not at the helm of this, antibiotic resistance or antimicrobial resistance or whatever, that is anti-drug resistance, for example, that can also be taken up.
Multi-drug resistance had been a big topic, and we have been consuming lots of drugs without even thinking twice.
True.
So that is altogether a different topic. I think today we will not be going there, because our main focus of discussion is systems biology and bioinformatics. So we will try to stick onto that area. Okay, this year 2024 we see Nobel Prize getting to Victor and Gary for microRNA and its probable role in gene regulation. I think I also saw you had some of your papers in microRNA as well as long non-coding RNAs. Right?
Oh, thank you so much, Som, for asking this wonderful question. I've been a great fan of Victor Ambrose. In fact, I had a great opportunity to hear to his keynote some 10 years ago. Gary as well, of course. And as I told you, I started my PhD career as a protein biologist working on hypothetical proteins. And then we discovered a large amount of microRNAs way back then, because some of those microRNAs were annotated as hypothetical proteins, and eventually we annotated those candidates as pseudogenes, actually. And then I've been very fortunate to have cited Victor Ambrose papers at least 40 times in my lifetime. And such a great honor that, I attended his talk and wonderful works. As far as microRNAs are concerned, there's still a long way to go. I mean, they didn't say that, okay, they've just discovered microRNAs that are probably play a very important role in regulatory mechanisms, but through their postdoc, they not only identified those LET4, LET14 mechanisms individually, and also they could drive the mechanistic evidence saying that, okay, these are those particular candidates that are probably regulatory in nature. And then their C. elegans work has given a large onus, a large amount of those particular times with respect to not only getting a large amount of data, large number of microRNA orthologs across different organisms, and then take that particular this thing to the translational value. So I think microRNAs have been a great great force behind understanding the regulatory this thing. I think it's been, I would still say that although this was discovered some 28 years ago, 27 years ago, they are a bit, I think they were nominated earlier 5-6 times as well, in my humble opinion, but it's been a bit delayed in getting their award, in getting them the award actually. Yeah.
True. Okay, so I think I have a very simple question, maybe for the students' perspective.
Right.
Is, we see systems biology as a top-down approach. So can you just explain that? What is this top-down approach is all about?
Yeah, a top-down is, taking a simple approach. Okay, you zoom in, you have got so many tissues, several tissues make up cells. So which means in humans we have 13.3 trillion cells. Now you further zoom in, 13.3 trillion cells, each cell has got a nucleus and you've got those organelles, okay? And then now you go to the nucleus where it has got its own genomic material, just like mitochondria, or in case of plants, you've got a chloroplast. And then when you go to the nucleus, you've got the chromatin that is supercoiled, and then you unfold it, and a bit of that particular euchromatin that is made up of DNA, which is double-stranded, and then you take up a bit of a DNA, then you define it as a gene, and then the gene transcribes and finally translates to protein. So that's where we are talking about the expressibility of the genome, okay, the mRNA to protein. So now when you have these particular proteins, at the end of the day, the protein represents how a phenotype is to be defined, how that particular individual can engross that particular characteristic feature that he has to do, okay. So that particular protein can only perform that function only when that particular protein interacts with another protein, be it a protein sitting next to it within the vicinity as a neighborhood, or a protein that is sitting far away. So this simplest correlation where a protein can interact with its neighboring proteins, and as a result we get to know the function of those particular proteins, is what we call it as a top-down systems biology approach.
Right. I think very well explained in very simple words. We usually, tend to use a lot of jargon, scientific languages, and make life of students a little difficult. I think from your explanation, whoever will be seeing this podcast, they will immensely get a very holistic idea what is this systems biology all about. So thank you, Prashanth, for giving me an explanation which I simply love it. That's I can say it for one line. Thank you, Dr. Prashanth, for your valuable time, and we agreed to come on this platform and share your views.
Thank you so much, for this wonderful organization. I mean, this has been a kind of a great podcast, and thank you so much, for the human service that you're doing, through Bioimagine as well.
Thank you, Dr. Prashanth, for your time and for wonderful, insight which you shared with our viewers and with me.